Hi everyone, welcome back to Module 2 of Chapter 3. So in this module, like I mentioned last time, we are going to talk about neuronal communication. So the first thing we're going to do is explain what neurons are, and basically the short answer is they are the building blocks of our nervous system. They are the cells that comprise our nervous system. And then we're going to talk about their structure as well as how they communicate with each other, including the chemicals that they use to communicate with each other, which are known as neurotransmitters, and in some cases hormones. So, Module 3.2 is how the nervous system works. So we'll talk about cells and we'll talk about neurotransmitters. So the brain, like the rest of the body, is made up of cells. There are two main types of cells that make up our brain. They are the neurons, which are the nerve cells, and then the glia, which are glial cells. So they actually serve to support the neurons functioning. So glial cells provide neurons with nutrients and insulation. They help them grow, they protect them from toxins and remove debris when they die. So they are very important, but the neurons are those that are considered the building blocks of the nervous system. They're responsible for sending and receiving messages throughout the body. And essentially, as we'll come to find out within the next few slides, their primary purpose is to fire. So neuron firing is how they communicate with each other. So you can see a little bit about the structure here. We have the soma or cell body. We have the dendrites, which are those little branch-like projections from the cell body. You have this long, kind of slender thing that goes away from the cell body. That's known as the axon. And then the little buttons at the ends of the axon are known as axon terminals or terminal buttons. So we'll talk a little bit about each of those. So there are three main parts that comprise each neuron. So we do have those dendrites that I was just talking about. They look like little tree branches. They act as antennas to receive messages from as many as 10,000 other nerve cells. And then they transmit those messages back to the remainder of the cell. So basically they take in those signals from the neighboring cells, and they're able to then kind of send them down that axon to the axon terminals and pass those impulses on to the next neuron in the line. So they form a chain reaction essentially, and we'll get into that in a little bit. Then we have the cell body or soma, which is shaped like either a sphere or a pyramid depending on which type of neuron you're looking at. And essentially, its responsibility is to include the nucleus, which holds the genetic information for the cell and controls the cell growth and reproduction. And it also contains biochemical machinery for keeping the neuron alive, so your mitochondria and things like that, as well as producing neurochemicals, so producing the neurotransmitters. And then finally, you have your axon, which transmits messages through what we call electrochemical reactions away from the cell body to other neurons or muscle gland cells. And they divide at the end into little branches known as axon terminals. And axons can actually vary from a few microns, so a very, very short length, to more than a meter in length. So some neurons, some nerve cells can have very long axons. So when the activity reaches the end of the axon and arrives at the axon terminals, it stimulates the release of vesicles, which are these kind of little bubbles containing neurotransmitters. So they are the chemicals that function as messengers, allowing neurons to communicate with each other. So although all neurons are designed to transmit information, they don't all perform the same function. So there are specialized different types of neurons. We have our sensory neurons, which receive information from the bodily senses and transmit it toward the brain, while the motor neurons carry messages away from the brain and towards the muscles. So the sensory neurons basically take in information from our senses, so what we're hearing or seeing or smelling or touching, send them up to the brain for processing. The brain decides what to do about them and then sends those signals back down through the motor neurons, which kind of control our muscles and our skeletal kind of system. So then we're able to move in reaction to whatever the sensation was. So again, I showed you this one at the beginning, but I wanted to show you it after explaining what the different pieces are. So you can see the cell body, you can see the dendrites, the axon, as well as the axon terminals, and you can also see a myelin sheath, you might notice in this picture, which is essentially kind of like a coating that covers the axon and helps speed the transmission of neural signals. And so I'll talk a bit in a minute about what that myelin sheath is made of and kind of how it functions, which is glial cells. So glial cells are specialized cells of the nervous system that are involved in mounting immune responses in the brain. So they help kind of protect the brain through immune system responses. They help remove waste, so any dead neurons, things like that, they will remove, get them out of the way. And they also synchronize the activity of the billions of neurons that constitute our nervous system. So they're actually very important. They're kind of like the coordinators for the neurons. Some glial cells form a white substance called myelin, which is basically a fatty sheath that insulates neuronal axons from one another, which is what I was pointing out to you a minute ago, the myelin sheath, which results in both increased speed as well as efficiency of neuronal communication. So constrictions within this sheath, so you can kind of see within the diagram, I can't flip back to it unfortunately in this mode, but you should see in the diagram, if you go back a few seconds, that there is kind of like little bumps within the axon. And so those constrictions, which we call nodes, divide the axon into different segments. So myelin sheaths prevent adjacent cell signals from interfering with each other and also speed up the neural impulses in general. So it makes the communication run a lot more smoothly. Unfortunately, damage then to our myelin sheath can lead to erratic signaling, as is seen in the case of multiple sclerosis. So you've probably heard of multiple sclerosis. It's a disease in which the immune system does not recognize the myelin and attacks it, and it unfortunately devastates the structural and functional integrity of the neurons and causes a lot of issues with functioning. So now we'll talk a bit about how neurons communicate, and this is probably as complicated as this chapter gets. I want you to kind of observe what's going on, keep in mind what's happening, try to kind of get the generalities, but I'm not going to be asking you to explain this process in detail because I recognize that it is very complicated. So neurons are not in direct contact with one another. There is something known as a synaptic cleft in between them, so it's a little gap in between the dendrites of one neuron and the axons of the other. And so this whole site, basically the terminal of one, the cleft, the gap in between, and then the receiving cell body and dendrites, is known as our synapse. So a single neuron may have many different synaptic connections. Neurons speak to each other using what we call an electrical and chemical language. So there is an electrical part of the kind of communication which occurs within the neuron itself, and then the chemical part actually occurs between the two neurons, so within that synapse, that synaptic cleft. So neurons at rest have a negative charge. So, and again, you don't need to memorize all these things, but there are more positive ions, so things like sodium, that have a positive charge outside of the cell than positive ions, for example, potassium, inside of the cell. And there's also a lot of chlorine inside of the cell, which is a negative ion. So basically what this accomplishes is that the overall charge of the inside of the cell is negative relative to the outside of the cell. And so this relatively stable state during which the cell is not transmitting messages is known as resting potential. So you can think of the word potential, it means that you kind of have the ability to do something. So when a cell is at rest, it's at resting potential because it's not actually doing anything at the moment, but it has the capacity or the potential to do something. So in this case, the negative charge on the inside of the cell is approximately negative 70 millivolts. So this seemingly stable resting state involves actually a great deal of tension due to the electrostatic gradient and the concentration gradient. So you can imagine if the inside is far more negative than the outside, those ions are wanting to move around naturally. So the electrostatic gradient refers to the fact that the inside and the outside of the cell have different charges, whereas the concentration gradient refers to the fact that different types of ions are more densely packed on one side than on the other. So for example, far more sodium outside, far more chlorine and potassium inside. So when a neuron is stimulated, so essentially when another neurotransmitter connects with it, the pores, known as ion channels, of its membrane open up and sodium rushes into the cell. So of course, if you have a negative resting state inside the cell and all of these positively charged sodium items start to rush in, ions start to rush in, you can see how the charge would be positively increased in this case. So when the charge reaches a critical level, which is known as a firing threshold, so for example, maybe it needs to get up to negative 55 millivolts from that original negative 70, something called an action potential occurs. So it's a wave of electrical energy that originates at the beginning of the axon near the cell body, because of course these dendrites are all connected directly to the cell body. And basically what happens is that that wave of electrical energy kind of originates there and rapidly travels down the length of the axon. And so basically what happens actually is in each of those little nodes along the way, you've got another little action potential occurring. So it's kind of like a domino effect. And so during your action potential, the charge changes from approximately the -70 of resting potential to approximately a positive 35 millivolts. But this change does not occur along the whole axon at once. Like I said, it does kind of have like a bit of a domino effect. Once the firing has occurred, there are mechanisms in place that return the neuron to its original resting potential so that it can fire again. So the ion channel slams shut, the sodium ions are manually pumped out because of course they're not going to want to leave on their own since they've now kind of started to restore that charge. And so removing the sodium ions, if you're kicking too many of them out, actively pumping them out, it can actually cause the cell to become hyperpolarized, which means that it is actually more negative than it would normally be for resting potential. And so this makes a cell less likely to fire and leads to what we call a refractory period. So this is a brief period of time in which the neuron cannot fire, so approximately 2 to 3 milliseconds. So until it climbs its way back up to that typical resting potential of, say, the -70 again, it is unable to generate an action potential within that point. So you have like a little bit of a period in which the cell can't fire. Now, of course, 2 to 3 milliseconds is nothing in terms of, you know, like our human kind of time frame, so we would not even notice this. But there is a point at which the cell can't fire for a few brief milliseconds. So when a neural impulse reaches the axon's tip, the message needs to be transported across the synaptic cleft, which triggers the release of neurotransmitters. So once it reaches the end of the cell, we know that since it's up until this point been an electrical form of communication, it can't just jump across the gap to the next cell. So that's where the chemical process comes into play. So the neural impulse travels down the axon, it kind of bangs into these little vesicles, which are full of neurotransmitter molecules that are located in those axon terminals, and it basically causes them to kind of pop and spill their contents into the gap, into the synapse. So it's important to note that neuronal firing adheres to the all or nothing principle. The individual nerve cells either fire or they don't. So there is no strength of firing. One doesn't fire any harder than the other. The main difference we'd see is in the frequency of firing. And so this is kind of an example of what we were talking about just then. So you can see at resting potential, we've got far more negative ions inside the cell and far more positive ions outside the cell. But then we see those positively charged ions starting to rush into the cell during an action potential, which again causes that charge of the cell to spike up from the -70 to say the approximately positive 35 millivolts. And then after the cell has fired, those positive ions are kind of pumped back out into the surrounding environment so that cell can now return to kind of that resting potential and get ready to fire again. And this is kind of a graphical representation of what's going on there. So again, if it's at -70 and slowly the ions are kind of coming in, and then it reaches, say, that threshold of excitation, we were talking about that threshold for firing of, say, maybe -55, and then all of a sudden, very quickly, that action potential occurs, right? So you see the really big spike up. It goes up, in this case, just past positive 40, and then it would kind of swoop back down as those positive ions are being kind of kicked back out of the cell, and then you can see how it might even return to a little lower than it was initially, which leads to that refractory period. So while it's slowly making its way back up to -70, it's not able to generate an action potential at that point. So neurotransmitters diffuse across the synaptic cleft, and they bind to receptor sites on the receiving neuron. So once those little, kind of that electrical charge has pushed those little vesicles to the end of the axon terminal, and they've burst and kind of spilled those neurotransmitter molecules into the gap, those neurotransmitter molecules are kind of free to cross that gap and to bind to the receptor sites on what we call the postsynaptic neuron, which is the one that's after the synapse. So they then produce changes in the receiving neuron's membrane, ultimately either acting to excite, which would shift the voltage positively, or inhibit, which would shift the voltage negatively, that receiving neuron. So depending on what type of neurotransmitter, they're either going to make that next cell more likely to fire. So if they're exciting it, if they're shifting it in a positive direction, then it's going to get more likely to fire. Or if it's an inhibitory neurotransmitter, it's actually going to cause it to become less likely to fire. So it's going to shift the voltage down negatively. So inhibition is very important. A lot of people would say, well, what's the point of having ones that will make it less likely to fire? But if you weren't able to stop your neuronal firing, you'd be unable to sleep. It would be very hard to coordinate movements. So you have to be able to kind of not only send, impulses that are going to create action, but also impulses that can allow you to slow down and stop. So once neurotransmitters detach from the receptors, they're either broken down or they go through what we call reuptake. So it's a process whereby neurotransmitter molecules that have been released into the synapse are reabsorbed into the axon terminals of the presynaptic neuron. So it's essentially a natural recycling process. So here is kind of a couple more little diagrams that kind of illustrate this. So the lock and key analogy for matching neurotransmitters. Essentially, neurotransmitters, when they cross that gap and bind to the postsynaptic cell, those dendrites of the postsynaptic neuron, they can actually only bind with receptors that are kind of specifically for that particular neurotransmitter. So it's kind of like a lock and key type mechanism. They only fit within the receptors for which they are designed. And then figure 3.14 here, you can kind of see how as the action potential, you see that red arrow is kind of traveling down the axon to the end. It kind of bangs into these little bubbles that we call synaptic vesicles, and they contain the neurotransmitter molecules. And if it pushes them to the bottom of the axon terminal, then they pop and spill the neurotransmitter molecules out into the gap, into the synapse, and then they are kind of they attach themselves to the little receptor sites on that postsynaptic neuron. And you can see the reuptake happening here. So after they've attached themselves to that postsynaptic neuron and created the desired effect, well, they don't need to continue staying attached to those receptors, so they kind of pop back off, and sometimes that neuron that released them will actually kind of suck them back up so they can be recycled and used again. So neurotransmitters make the excitation or inhibition of neurons possible. They exist in the brain, the spinal cord, the peripheral nerves, as well as certain glands within the body. So essentially, they control everything your brain does. The nature of their effect depends on their particular location, so what brain area they're located in, as well as the type of receptor it binds to. So like we said, there are specific receptors for specific neurotransmitters. There are two well-known neurotransmitters which exist throughout the brain. There's something known as GABA, or gamma aminobutyric acid, and it's the primary inhibitory neurotransmitter of the nervous system. So it's the one that causes a receiving neuron to be less likely to fire again. And so essentially it is produced by a lot of cells in your brain. It is the most common inhibitory neuron that we have within our nervous system. And then we have glutamate, which on the other hand is the most common excitatory neurotransmitter in the brain of vertebrates. And we are vertebrates. Vertebrates mean having a backbone. So it is released by about 90% of our brain's neurons. So obviously very, very kind of prevalent and common in the brain. And so it is kind of the primary excitatory neurotransmitter that we have within our nervous system. Some other major neurotransmitters include serotonin, which you've probably heard of before. We tend to use drugs known as selective serotonin reuptake inhibitors to treat conditions like depression and anxiety. And those basically have their function by preventing the serotonin from being kind of reabsorbed by the presynaptic neuron, which causes there to be more serotonin available for use within the brain. So serotonin is a monoamine involved in sleep, appetite, pain suppression, temperature regulation, mood, aggression, and sensory perception, just to name a few of its common features. Dopamine is another very common neurotransmitter that we talk about a lot, especially in the realm of clinical psychology. It's a monoamine involved in voluntary movement, memory, mood, attention, learning, and pleasure and reward. So it actually mediates the pleasure and reward response system within our brain. and it's possibly implicated in response to novelty, so kind of new situations as well. Norepinephrine, maybe something you've heard of. We have norepinephrine and epinephrine. And if you've ever heard of an EpiPen for somebody who has a severe allergy, for example, myself, that's actually, an epinephrine is actually kind of a derivative of this chemical. And so basically it's involved in regulating stress responses. It helps slow intestinal activity, increase heart rate and respiration. So it can be involved essentially in that kind of fight or flight response. So it's essentially kind of an adrenaline. So it's involved in learning, dreaming, waking from sleep, memory, and emotion. And then finally we have acetylcholine, which is one of the most widespread neurotransmitters within our nervous system. It's found at the junctions between nerve cells and skeletal muscles, so basically where the motor neurons are talking to the skeletal muscles to tell them how to move. And it affects cognition, memory, and emotion, as well as some other things. So you don't have to remember every single detail about all of these, but you should be kind of familiar with what some of their key functions are. So if you can kind of keep in mind a little bit about what's here in this table, that should be pretty much enough to kind of know with respect to the different neurotransmitters, the major ones which we talked about. So if neurotransmitters are too high or too low, they can have harmful effects. Abnormal GABA levels, that gamma aminobutyric acid, are implicated in sleeping, eating, and convulsive disorders. Alzheimer's has been linked to acetylcholine levels, and dopamine levels are linked to Parkinson's disease. So we can see how some of these neurotransmitters, having too much or too little of them, can lead to some really adverse conditions. However, pinning down the exact relationship is very tricky. So each neurotransmitter plays multiple roles, and their functions often overlap. The effects could actually be in the opposite direction of what we think they would be. So even though we know that two are linked, it doesn't necessarily mean we know the direction. So as we mentioned in chapter 2, and we've mentioned several times since, correlation does not equal causation. Even though treatment that boosts or decreases neurotransmitter levels may be effective, this does not mean that levels of the neurotransmitter are what actually cause the disorder. So that's kind of like the whole aspirin and headache conundrum, which you may have heard before as an example of kind of how we can't really establish causality in that way. So for example, if we give someone aspirin and it relieves their headache, does that mean that the headache was caused by lack of aspirin? Probably not, right? So even though if we use, say, treatment that boosts serotonin to treat depression, so the SSRIs I was just talking about, that doesn't necessarily mean that it was a lack of serotonin that caused the depression in the first place. So we still can't really establish causality. We just do know that some of the treatments using these neurotransmitters can help with particular disorders. We regularly ingest things that affect our neurotransmitter levels, so things like recreational drugs, herbal remedies, even some ordinary foods. They contain, things that will affect our neurotransmitters. And the relationships and interactions are complex. So really, there's not one-to-one relationships. We can't really establish what causes what, but we have found some treatments with neurotransmitters to be effective for certain disorders, and that is a bonus for us because it allows us to help relieve symptoms for people who suffer from some really adverse conditions. So here's a little kind of diagram of the drug effects on neurotransmitters. So drugs can affect the chemical signaling between nerve cells. If we have an agonist, so any drug that is an agonist, it enhances or mimics the effects of a neurotransmitter's action. So for example, nicotine, it will actually mimic the effects of particular neurotransmitters. Whereas antagonists inhibit neurotransmitter activity by blocking receptors or preventing the synthesis of the neurotransmitter. So for example, something like Botox, which is actually derived from a toxin, which it's really interesting. People inject this into their faces to look good. It's also used to actually treat some medical conditions as well, but a lot of people use it for cosmetic purposes. But essentially what it does is it actually inhibits your muscles in that particular area from moving. So it's an antagonist because it's inhibiting the neurotransmitter activity. It's blocking those receptors so you can't have motion within those areas, which is why it kind of basically paralyzes you. So yeah, your agonists are always going to basically enhance or mimic the effects of a particular neurotransmitter, whereas your antagonists if you think of someone who antagonizes somebody, they're kind of like the bad guy, right? They kind of block something. So they inhibit the neurotransmitter activity by either blocking the receptors or preventing the synthesis. And then finally, I want to talk briefly about hormones, because hormones can actually also affect our nervous system functioning as well. So they're actually chemical substances secreted by glands of the endocrine system, which is kind of a different bodily system, but very closely related to a lot of our other systems. They're released directly into the bloodstream, so they have to be carried to organs far away from where they originate, so they can be carried to organs far away from where they originated, and they have dozens of jobs. So whereas with neurotransmitters, they kind of hop from one neuron to the other, so they really only exist kind of within that nervous system. The hormones are released into the bloodstream, so they can kind of travel in that way. So it's important to note that neurotransmitters and hormones are not always chemically distinct. So sometimes the same substance can act as both a neurotransmitter or a hormone depending on where it's located within your body. So norepinephrine, for example, when it's released, within your nervous system, we would consider it a neurotransmitter if it's being kind of transmitted between the neuron cells within our nervous system, our nerve cells. But it can also be secreted by particular areas of your endocrine system as well and released directly into the bloodstream, in which case it would be acting as a hormone. So there are 4 types of hormones that are of particular interest to psychologists and neuroscientists. These are melatonin, which you've probably heard of, oxytocin, adrenal hormones, and sex hormones. So melatonin is secreted by the pineal gland, which is a small gland within your brain, and it helps regulate biological rhythms and also promote sleep. So we know if you've ever heard of somebody taking melatonin to help them sleep, that will be why, because it's actually involved in regulating our sleep patterns. We also talk a lot about oxytocin. So oxytocin is actually primarily implicated in attachment. So it's secreted by the pituitary gland, which is the master gland of the endocrine system, which again is also located within your brain. It produces hormones and sends commands about hormone production to the other glands of the endocrine system. So the pituitary gland is kind of like the boss of the whole endocrine system. And so it is the one that produces this oxytocin, and oxytocin can enhance uterine contractions during labor. It facilitates the ejection of milk during feeding, breastfeeding of a child, and it also contributes to relationships. So like I said, it has been highly implicated in attachment and kind of forming relationships and bonds. Specifically, we talk about it a lot with the attachment between a mother and child, because as we just mentioned, it was secreted during childbirth as well as during breastfeeding, but also it can be implicated in romantic relationships and other relationships as well. Then we have our adrenal hormones, which are produced by our adrenal glands, which are little glands that are located kind of adjacent to our kidneys. And they are involved in emotion and stress primarily. So we'll talk about that. If you continue on to the second course with me, we have a whole chapter on kind of stress and health, and we'll talk a lot more about how there are different pathways for a stress reaction. And so essentially, the adrenal hormones are involved in emotion and stress. They include cortisol, which is a hormone that increases blood sugar and boosts energy, so kind of gets you prepared to deal with a potential threat, so something that's stressing you out. We have epinephrine, which is adrenaline, and norepinephrine, which is also, again, kind of like a form of adrenaline. And again, so these kind of relate to both the nervous system and their use as neurotransmitters, but also they are produced by the adrenal glands, which are a part of our endocrine system, and in that case, they would act as hormones. So they both act to increase your arousal and prepare you for action. So again, if you're talking about that fight or flight response that we have when we're faced with a potential stressor or a threat, that would be largely produced by the effects of things like epinephrine and norepinephrine. And they've also been implicated in memory. Again, you don't need to remember every detail about them. This is kind of just an overview of some of the most important ones. Then we also have the sex hormones, which are secreted by tissues in the gonads. So gonads are essentially our kind of our organs that produce, those reproductive cells, so our sperm cells and our ovum and ova, and that would be the testes and the ovaries, and also by the adrenal glands as well, and they include 3 main types. We have our androgens, which are our masculinizing hormones, so things like testosterone, and we have our estrogens, which are our feminizing hormones, which would contain estrogen and progesterone, which you've probably heard of just in the context even of things like birth control. A lot of forms of birth control will actually contain synthetic versions of estrogen and progesterone. And testosterone, I'm sure you've heard of, in terms of, it's largely implicated in a lot of male-specific behaviors. Females have testosterone as well, but not as high amounts as you would see in males, typically. So endorphin is another major hormone that's mentioned in the text. It's produced by pituitary gland, which I kind of mentioned earlier, and the hypothalamus, which is also another little area in your brain that's part of the endocrine system. And it functions to reduce pain and induce feelings of pleasure. So you've ever heard of an endorphin rush? Feeling really great. Some people say they get what they call like runner's high if they're exercising a lot, and like they kind of all of a sudden get really into it and they start to feel really good, and that's caused by the release of endorphins within the brain. So the actions of the pituitary gland are stimulated by another little brain area known as the hypothalamus, which is a brain structure that regulates basic biological needs as well as motivational systems. And that is the end. So I'm not going to get into any more detail, as long as you can kind of remember some of those examples of different hormones and neurotransmitters and a bit about what they do, as well as trying to kind of grasp the basics of that neuronal communication, how they kind of communicate between each other, and how that communication is both electrical and chemical in nature. That's kind of the main things that you need to take away from this module. So a bit of a shorter lecture than the last one. The next one is pretty lengthy. It's going to be the actual structure of the nervous system, so we'll talk about the different divisions of the nervous system as well as all the different brain areas. So that one's definitely going to be a little bit heavier than this. But again, like it's mostly memorization type stuff. It's not going to be super hard to understand. It's just like memorizing the different areas and kind of what they're related to. So I will end this here for now, and I will see you in the next video.